Hi everyone, welcome to Bio 6001, Advanced Cell Biology and Bioimaging. I sure love talking about bioimaging, so just wave your hands at me if I'm going on for too long. We're going to do a lot of cell biology in this module, but the key thing to start with is the advanced bioimaging component of it. Now, in a normal year, you would get into the Center for Bioimaging at Oxford Brookes and you'd get your hands on all of our big microscopes, confocal microscopes, electron microscopes, and we would do our practicals that way. We can't do that this year, at least for the start of semester, so it's important, and what I'm going to do for the first couple of weeks is tell you all about different advanced imaging technologies and techniques. So it won't be until week 3 that we actually get to talking about cell biology a little bit. Okay, let's move on. First thing is the distinction between light and electron microscopy. The microscopes that you use in the lab to look at microscope slides are light microscopes, but equally so are the big fluorescence microscopes and the big confocal microscopes that I'm going to tell you about. Any microscope that uses photons as a source of light to illuminate the image and produce the final image is a light microscope. So there are different categories: bright field, fluorescence, and confocal under light microscopy. Then, of course, there's electron microscopy. By the way, in the Center for Bioimaging, they're divided in different rooms. I spend a lot of my time in the light microscopy suite. Don't very often get across the hallway to the electron microscope room anymore, but I started out my career as an electron microscopist. And there are two different flavors of electron microscopy, if you will. Transmission electron microscopy, there's an image there generated with a TEM. And then there's scanning electron microscopy, which makes these beautiful 3D surface representations of organisms. So for this week, I'll talk about light microscopy, some of the techniques that we use in the various light microscopy applications. And then next week, I'll get more into the experimental light microscopy and the electron microscopy. So what's the real difference between these two things? Light microscopy works at lower magnification than electron microscopy does. It still makes very beautiful images, as you can see, but critically, it can let you look at living cells. And this is, there was a complete paradigm shift in biology, and cell biology in particular, about 10, 15 years ago. A little earlier than that, maybe. But people started looking at living cells. We'd done everything we could looking at fixed section samples. It was time to start looking within cells, looking at organelles moving around, look at how proteins associate with each other, that sort of thing. So you do that with light microscopy. Electron microscopy lets you look at things at incredibly high magnification. You can go right down inside the cell and you can look at the details of membranes and things like that, but you've got to kill the cells before you do that. They have to be fixed with a chemical. They have to be embedded in a plastic or a resin. They need to be sectioned and stained so that the electrons bounce off the membranes. And if you could invent an electron microscope that would let us look at living cells, oh, that's like the holy grail. That would be a complete game changer for cell biology. Two concepts that we've got to talk about before we get to looking at the actual microscopes: magnification and resolution. Magnification is kind of an easy concept, I think. It is the size of the image you produced divided by the actual size of the object. If I take a photograph of you, click, and I blow up the picture of your face so that it covers the side of a building, well, all you have to do is multiply how big that, or divide how big that is by the size of your actual face, and that's the magnification. Maybe it's 100 times, maybe it's 1,000 times, what have you. So I'm not going to talk too much more about how that's accomplished just at the moment, but I do want to talk about resolution, the fineness of detail that can be distinguished in an image. Here, we'll do this as a thought exercise. Two incredibly small objects, maybe they're organelles inside of a cell, at low resolution will appear as one big blurry object. At high resolution will appear as two distinct dots. So our goal in advanced bioimaging is resolving power. We want microscopes that can give us the best resolution possible at high magnification. And let's look at typical resolutions of microscope systems. Light microscopes like fluorescence, confocal microscopes will let us resolve two objects that are within 200 nanometers of each other. It'll let us see detail down to 200 nanometers in size. You've got to think about your SI units. If you look at a ruler, the smallest division generally on a ruler is 1 millimeter. Divide the millimeter into 1,000. That's a micrometer. That's incredibly tiny. Divide that incredibly tiny micrometer into 1,000. That's a nanometer. 200 of those nanometers, 1/5 of a micrometer, is what the best resolving light microscopes can see. We have a new category of microscopy just recently called super resolution microscopy, which uses tricks to resolve even better than that theoretical resolution limit of light microscopes. I'll talk about super resolution microscopy techniques next week, but then look at the electron microscope. 0.1 of a nanometer, 1/2000th of what the light microscope can resolve. So the electron microscope is a beast. It can do things at incredibly high magnification, and it can resolve incredibly fine detail. Let's give you a real-world example of resolution. In a very well set up, very high-end research microscope, I want to look at the Golgi bodies in a plant cell. Those little green dots that you see running around are Golgi bodies that have been stained with a green fluorescent protein. And I love watching them. You can watch them move around for hours. This is quite highly magnified. Each of those Golgi bodies is about 2 micrometers in diameter, and you can see them quite clearly, but you don't really see any detail of their internal structure at all. If we fix this tissue, embed it, section it, stain it for transmission electron microscopy, we can easily see the internal details of the membrane systems within the Golgi apparatus. And we'll talk more about the biology of the Golgi as the module goes on. So, lower in resolution and magnification, but able to view living processes, much, much higher in magnification and resolution. And just in case the concept of resolution hasn't quite gotten into your brain, what's that a picture of? You might be able to have a guess by looking at it. A low resolution image, 72 pixels per inch. Can you make out what it's showing? I think I'm biased because I already know what it is. Do you know what it is? You might be able to make out what it is by looking at that picture there, but that would be said to be a very low resolution image. You can see that it's made up of square blocks. Those square blocks are called pixels. And a pixel in computer storage can only have one color, which is why we get these blocks of color. If we look at a high resolution version of that same image, well, you knew it was a water lily flower, I'm sure, but you couldn't really count the petals in here, could you? You couldn't see the details in this water down here. Look at that, absolutely superb. This is a 300 pixel per inch image. So each one of the pixels here has been divided into 4 that way and 4 that way. There are 16 pixels where there was 1 pixel in the image here on the left. 16 pixels can all have different colors, which is what gives you the fineness of detail, which is the higher resolution that we're talking about. So we strive for high resolution and high magnification in our research microscopes. And how do we get that? We get that by paying a lot of money. The microscopes in the bioimaging center cost hundreds of thousands of pounds. The microscopes on the lab on the bench in the teaching lab cost hundreds of pounds. There's a big difference between the two things. The objectives in the confocal research microscopes can cost 5,000, 7,000, even 10,000 pounds. The higher the price, the higher the resolving power of the objective is the general rule of thumb. I'm going to show you some maths, but don't worry, there's not a whole lot of that in here. Given that I'm giving a lecture on optics, there's not a whole lot of maths at all. You look at this equation here. This is called the Rayleigh criterion, or the Rayleigh equation, where D stands for the minimum resolving distance. You want D to be as small as possible. Well, if you've got an equation that has this structure here, if you make this number here bigger, then that number there, the minimum resolving distance, gets smaller. So that would be our goal, to make n sine theta larger. N sine theta You look down here is a quantity that's called the numerical aperture of the objective being used. And here are three objectives drawn in this picture. This one here has a low numerical aperture of 0.25, medium. This one here has a quite high numerical aperture of 0.95. If you look critically at the picture, you can see that this one gathers a relatively narrow cone of light from the specimen, whereas this one here gets really close to the specimen and it gathers a wide cone of light. It gathers lots and lots of light from the specimen. That's where we get the increased resolution that I'm worried about. So it is that angle there, theta, that determines the minimum resolving distance of an objective. You don't really need to worry what the individual terms represent, just that numerical aperture is the bottom thing in that equation. When you look at, physically take an objective from a microscope and look at what's printed on the barrel of it, it'll say the magnification. It might say 40x or 63x or 100x, something like that. But right underneath that, there's another number. It'll say 0.8. or 0.95 or 1.2 if it's a really high numerical aperture objective. So when I gave a lecture about imaging right at the start of first year in cell biology and genetics module, I talked about basic light microscopy, and I'm not going to do that again with you guys. I'm going to jump right to fluorescence microscopy because it's what we primarily use in research in cell biology. So fluorescence microscopy, several different aspects to discuss in this lecture. The principle of fluorescence, the kind of equipment we use, and fluorochromes, the kind of stains we use in fluorescence microscopy. Then I'll spend some time talking specifically about confocal microscopy, which is a subcategory of fluorescence microscopy. And then I'm going to talk about fluorescent proteins in the second half of the lecture today. Of course, it's all well and good to make pretty pictures with microscopes. You can put them up on the wall, you can amaze your friends and family, but to justify spending all of those hundreds of thousands of pounds on this equipment, we've got to be making scientific breakthroughs. And that's our bread and butter. That we do the research, we discover things, we publish papers, and we're recognized as doing cutting-edge science. So that, of course, there are all sorts of experimental techniques that let us do things like measure protein interactions. That's my symbol, by the way, for protein interactions. You're probably going to hear that a lot this year. So next week's lecture will concentrate more on the experimental techniques that we do with light microscopy. First of all, what is fluorescence? All matter is made up of atoms, of course, and if you excite an atom by inputting energy into it somehow, its electrons jump from ground state, so-called. The electrons in the outer electron shell jump from ground state out to a higher orbital. I don't know if you took chemistry and remember all of the details of this. Don't worry, I'm not going to test you on it. But an input of energy will cause atoms or molecules to get excited. Here's a fine example right here. I just heated my coffee up in the microwave. It's nice and warm. It's nice and warm because the microwave energy has excited all of the atoms in the coffee. They've jumped up to that higher energy state. They don't stay there very long, though, in that high energy state. They fall back to ground state. And when they fall back to ground state, they do it by releasing heat. So all atoms do that. All matter does that. Here, if I rub my hands to get them warm, what I'm doing is I'm putting energy into the atoms, and it's generating the heat of friction that you feel in your hands. Heat is emitted. There are some molecules that have a very special property, and that is that they are fluorescent. When they fall back to ground state, they do emit some heat, but they also emit photons, which are light. Molecules that come in that category are called fluorochromes. So you put some energy in, usually in the form of light, and when they fall back to ground state, you get light back out. A typical fluorochrome has what's called an emission, an excitation spectrum, and an emission spectrum. So if you look at the x-axis on this graph here, this is visible light from 300 nanometers all the way out to 700 nanometers. Visible light really starts at about 400. Down at this end we have ultraviolet radiation, and out here we have infrared radiation. So we're talking about light that's in the visible spectrum, light that we can see. This fluorochrome, this fluorescent molecule that I'm talking about here, whichever one it is, seems to have a real peak of excitation in the blue light range at about 480 nanometers. If you shine blue light on this molecule, it gets excited, it emits light, and the light that it emits has a peak wavelength in this green region here at about 530 to 540 nanometers. So that is very common for a fluorochrome. Excitation peak, emission peak. The widths of these peaks can vary. The number of peaks that a different fluorochrome has for excitation or emission can vary, but in practice we use ones that look like this. A sharp, well-defined excitation point and a sharp, well-defined emission point. Emission, the fluorescence that you see when you excite a fluorochrome, is always at a longer wavelength. It's always down this way towards the red. That's because light energy that goes towards the red has less energy. Light that's down here in the UV range is much more energetic than light that's down here in the red range. So put energy in, get some lower energy out. If it was the other way, if the emission was down here, we'd actually be creating energy, and we can't do that. That would solve all of humanity's energy crisis issues, but we can't do that. So that's fluorescence. Now, if you're looking at a fluorochrome, in this case here, the example fluorochrome is excited in the UV light range, and it emits its fluorescence up in the blue range there. But if you stain something with that stain, and you tried to look at it with your microscope, what would happen is you'd be shining UV light down on the specimen, looking through the eyepieces, and the UV light would be going into your eyes and blurring out, washing out the blue image that you want it to see. So a fluorescence microscope has to have a special third piece of glass, and that's this thing here called a dichromatic or dichroic mirror is what I call it. And the dichroic mirror allows the excitation light to hit the specimen, but then it blocks it from coming to the detector, which might be your eye or it might be a camera of some sort. So we need 3 pieces of glass filters in a fluorescence microscope, and those in modern microscopes are arranged in cubes like this. We call that a filter cube. It has the excitation filter, it has the dichroic filter, and it has the emission barrier filter. And the barrier filter, by the way, lets light in a certain wavelength range through. They can be very narrow. We might just want to detect the light just there at that peak. So if you've got a fluorochrome that emits in the blue region, you would want to use one specific filter cube set up. If you've got one that emits in the green region, you would use a different filter cube. One in the red region, a different filter cube. The modern microscopes have many, many different filter cubes, and you just spin the one into place that you want. Or in fact, you push a button and the turret automatically rotates to the one that you want. And so in very short order, we've come to confocal microscopy. Fluorescence microscopy is the big umbrella topic here. Confocal microscopy is a subset of fluorescence microscopy, but it's a really big and really significant subset. And in fact, most research that's conducted in cell biology these days is done with confocal microscopes. They allow you to make, well, absolutely stunningly beautiful, clear images of biological specimens in all sorts of fluorescence colors. Let's go back. Let's do a bit of a history lesson here. This guy, Marvin Minsky, I see I cut his first name off there. Poor Marvin. Marvin Minsky invented the confocal microscope while he was a PhD student at Harvard University way back in the 1950s. But confocal microscopes weren't made commercially until the 1990s, 40 years later. And that's because he was a smart guy. He was a physicist, and he could draw these things on paper, and he could do them as thought exercises. His PhD examining committee read his thesis, and they said, yeah, that works. That's absolutely brilliant. They made him a doctor, he became Dr. Marvin Minsky, and over his illustrious career, he went on to co-found the Massachusetts Institute of Technology's Artificial Intelligence Department. I mean, he really knew his stuff. But anyway, his PhD examining committee said that idea for that microscope that gives a super clear image, that really sounds good, but they couldn't build it. And the reason they couldn't build it, well, will become clear. Let's move on. So here are confocal microscopes. These are here the two confocal microscopes that we have in the Bioimaging Center at Oxford Brookes. This picture here was taken a few years back. This is one of our old confocal microscopes. I still think of it as the new one, but we've got even newer ones since then. I love this picture because it reminds me of a time I used to work with these two guys, and I often wonder where they are now and what kind of great scientific research they're doing. Anyway, they've got a microscope slide there on this confocal microscope, and they're making a beautiful image. You can just see it appearing on the screen here. The key things that a confocal microscope needs, well, it needs to be a research microscope equipped as for fluorescence. That's a fancy way of saying it needs to be a fluorescence microscope. Not, oh, by the way, not a fluorescent microscope, as a lot of people write when they're writing essays in this module. A fluorescent microscope would be one that was glowing. It's a fluorescence microscope. Try to remember that when you're creating practical and essay coursework writing. A confocal microscope needs lasers of various output wavelengths, and that's what all modern confocal microscopes have. But in principle, you could do confocal microscopy using a light bulb like in a normal fluorescence microscope, but we don't. We use lasers. That'll become clear later on. I'll explain why. They also need a scanning mechanism because the laser, think about a laser pointer. It's a really teeny tiny dot of light. It needs to be scanned back and forth across the specimen so that the image builds up on the screen. You need light detectors and amplifiers. I will explain that in the fullness of time. You need a computer with substantial processing power. Now, in the old days of confocal microscopy, if I think back 15 years, I'd be trying to make a series of images, maybe 100 images of a biological specimen, and oh, the computers just could not do it. They would get 30 or 40 images stored in their brain, and then they would start to slow down, and oh, eventually the computer would crash. It was horrible. Computer processing power has increased exponentially since then. The confocal microscopes that we have nowadays can record 500 to 1,000 images in their brain, maybe more, while you're doing other things on them, while you're analyzing other data sets. It's just incredible what the computers can do these days. And you need suitable fluorochromes. That means all of those different fluorescent molecules that I was talking about. You need fluorochromes that emit in the correct color region so that you can stain the different things within the cells you're studying. Why didn't Minsky build a confocal microscope? They had fluorescence microscopes in the 1950s. Lasers, I'm not sure. I certainly wouldn't think they had lasers in all the suitable wavelengths and that sort of thing. But I told you earlier that you could, in theory, do confocal microscopy with a regular white light bulb, so that wasn't the problem. Scanning mechanism wasn't required if you weren't using lasers. I'm going to skip over this one for a minute. Computer with substantial processing power didn't exist then, but the original confocal microscope would only have made a single image, and that could have been recorded on photographic film, like all images in biology were done right up until the 1990s. Anyway, so also not an issue, and they had loads and loads of fluorescent molecules even in the 1950s, so not a problem at all. Light detectors and amplifiers were the problem. These are electronic devices that detect photons. Photon hits one and it amplifies a single photon so that it becomes something that you can see in an image. Your eye can't see a single photon. It can't even see a very small quanta of photons. It can't see a small group of photons. What am I trying to say? Your eye needs a lot of photons before it registers that there's light coming into it, okay? But the amount of light that comes through a confocal microscope is very, very low, and it needs to be amplified. So that sort of electronics there needed to be invented still in the 1950s. And along with the advent of laser technology, it's those two things together, I guess, that really meant by the time we got to the 1990s that we had the technology in place to make confocal microscopes. So I'm banging on about it, but how is a confocal microscope different from a conventional fluorescence microscope? This picture here is showing what goes on light pathway wise within a conventional fluorescence microscope. And if we look down here, you've got to pretend that your specimen is relatively thick and three-dimensional, and it's sitting down here maybe on a microscope slide, which would be at the bottom of the picture. Now, you guys already know from looking through microscopes that you can focus up and down through your specimen. If you focus up and down through a specimen that's stained with fluorochromes, you find it, you get a good focal plane, you think, okay, I want to take a picture right at that focal plane there. What happens is, here, I'm just going to magnify this region down here where the so-called focal plane is. So here is your specimen, the big yellow blob here, and you're looking at it from up above. You want to focus right on the middle of the specimen here. See the green lines that are converging? That's the light that's being emitted right from the focal plane. That's the thing that you want to see within your specimen. But what happens when the fluorescence excitation light is coming down through the specimen? You're also exciting fluorescence up here above your focal plane, and you're exciting fluorescence down here below your focal plane. All of that light goes traveling back up through the objective of the microscope, gets focused by a lens into the detector, which might be your eye, or it might be something like a photomultiplier tube. So you see your focal plane, but you also see all of the out of focus blur. And it means that the image that's created by a fluorescence microscope isn't super satisfying. I'll show you an example just in a minute. So let's go to the next slide and look at what Minsky came up with, the confocal principle. I'm amazed that he got a PhD from Harvard University for doing this, but it was a stroke of genius at the time, I promise. Minsky put a little aperture right in here, a little plate, a little metal plate, and in it he put a little teeny tiny hole that he called a pinhole. So the pinhole is the critical thing in confocal microscopy. Look what the pinhole does. All of these light rays are converging on the detector. Remember, the green light was being emitted from the focal plane that you want it to make an image of. It focuses right at the center of the little pinhole, and it goes into the detector. And the detector forms the image. The out of focus light that came from above the intended focal plane gets blocked by that aperture, and the out of focus light that came from below that focal plane gets blocked by the aperture. So none of your out of focus light gets into the detector, which means that you get a really, really sharp, clear picture from right here at this level within your specimen, and you don't get any of this up here or any of this down here. Genius. And it really, really works in principle. Wait till you see the pictures. Pinhole is very, very small. I say less than 100 micrometers in diameter. If you make a piece of cardboard and put a little pinhole in it and then try and look at a light source through it, you'll see that not very much light comes through. And that's why I was saying that you need to have the photo amplifiers that will amplify that signal into a visual signal, into an image on your computer screen. Here is a picture of a pollen grain, and the pollen grain has been stained with two different fluorochromes, a red stain on the outside, and it's got a bright green stain inside of it. But when we image it with a normal fluorescence microscope, we just see this blurry out of focus red blob. When we image it with a confocal microscope, we can focus down into the middle of the pollen grain, and we can see that it has the beautiful red fluorescence around its outside, and it's got lots of bright green fluorescence inside. So I'm focused here somewhere down in the middle of the pollen grain, But the reason I'm showing you a series of images is because, of course, you can change your focus. You can go up above the pollen grain and you can make a picture. You don't see any of the stuff down below. You can step down a little bit, step down, keep stepping down all the way through the pollen grain and make what's called an axial series going down through your specimen. In confocal microscopy talk, we call that a Z series. X and Y are the two planes of a two-dimensional image, and the Z axis is going straight down through the image that way in a three-dimensional image. So we've got all of these individual sections, much like the brain scans that you see that are done by magnetic resonance imaging. They go up and down through. Remember, the key thing here is that this is all completely non-invasive. We're not cutting the pollen grain physically. We're just using the confocal microscope to look down through it and show us all of these beautiful details inside of it that you can't see with the regular fluorescence microscope. And I've already given it away down here. Once you have all of these beautiful, beautiful clear images of the different focal planes, you can use computer software to do what's called a Z projection, an axial projection, and you can see a very, very beautiful 3D surface rendering of the pollen grain with much, much more detail than what you could see here. Show you a real-world example of this. This is a living plant cell. Well, it was living when I made the movie. I don't expect it's still living. If it is living, it's the longest-lived tobacco plant of all time. What we're doing is we're looking at a single cell, part of a single cell. I'm just going to stop it. Looking down on the top of the cell, I've got the endoplasmic reticulum in this cell marked with green fluorescent protein, which we'll talk about in a bit. And I've got the Golgi bodies marked with red fluorescent protein. Sorry, the pointer's driving me crazy. There, it should stay now. And as I use the confocal microscope to step down and make a series of images, you'll see something peculiar happen. Let's play the movie. In the top image, here, you can see the endoplasmic reticulum, but then it disappears as we go further down through the cell. What's going on there? Well, I mean, all the people that just finished taking the plant science module with Green Planet will be yelling at the computer screen right now saying, oh, it's the vacuole in the cell. There's nothing inside of it. You'd be quite correct. All of the endoplasmic reticulum occurs just out at the periphery of the plant cell. But still, when I look at this series of images that we're stepping down through, I can't get a really good three-dimensional sense of what the top of the plant cell looks like. But of course, you can make the three-dimensional projection of that I was talking about on the pollen slide a minute ago. And we stop that. You can see really, really beautifully the endoplasmic reticulum structure in this cell. You can see the little red dots that are the Golgi bodies. That's all well and good, but there's something else that you can do with confocal microscopy, and this is the real wow factor thing for me. When I have you all in the bioimaging suite and we're doing confocal microscopy, we do these so-called Z series, and then we make these projections, and people go, oh wow, that's very cool. But then I do this. Confocal software recognizes depth in the Z direction because it knows where every pixel of light information is coming from. So what it can do, as well as projecting the image like this, is that it can compute rotations of the specimen that have been optically sectioned. And I'll just show you a movie of that. Look at this. You can really start to see the three-dimensionality of the endoplasmic reticulum. You can see that the surface of the leaf is slightly curving. So from individual optical sections to a 3D projection to a 3D rotation, all from a living sample. I could have, when I finished, taken this little seedling off of the microscope slide, put it into some dirt, and grown it so that it produced the next generation of seeds. I don't think I did that in this case, but I could have, and I have done with other specimens that I've imaged. So it's completely non-damaging, non-destructive, non-invasive. And that's the end of the first part of today's lecture. There's a second part of today's lecture. If you go back to Moodle, you'll find week 1 lecture part 2.